Hello everyone, welcome back to Chinmoy's Biology channel to a new episode that is part 2 of how do organisms reproduce. So we have already gone through the first episode. If you have not gone through that, I'll be putting the link in the description box as well as in the I link above. So please go through that, then it will be easier for you to understand this one as it is a continuation of the previous lesson. So let's get started with today's episode, and we will mainly be dealing with the asexual reproduction. And please stay tuned till the end because it will be a very important episode. As I always keep on telling you, don't skip in between because throughout the episode there will be some of the other important things which will be introduced to you in this class itself. So let's get started. So when we are dealing with asexual reproduction, first type of asexual reproduction, I told you in the previous class, we will be dealing with the binary fission. So what is binary fission? So if you are going through your NCERT book, you will see that they have generally given an overview of binary fission and all, but we should study them in details because you are in 10th grade and in your board exams you will get these questions in different forms. So we will go through it in detail. So what is binary fission? Binary fission is a type of asexual reproduction in which a single parent cell divides into two identical daughter cells. It is most commonly observed in unicellular organisms, right? And like bacteria, amoeba, and paramecium. Is it clear? Now in binary fission, the parent duplicates its genetic material DNA. Okay, we all know that the genetic material is DNA. It is duplicating the genetic material and the cell grows in size. The duplicated DNA separates. Okay, the cell is growing in size and the duplicated DNA this separates. The cell constricts in the middle and eventually splits into two equal halves. So the cell is constricting in the middle and it is eventually splitting into two equal halves and with a copy of the original DNA. So one copy of original DNA and one copy of the formed or the new DNA they combine, and these two daughter cells are genetically identical to each other and to the parent as well. It's clear? So this is how binary fission from the parent, it duplicates the genetic material to the daughter cells. They grow in size, then the genetic material it's divided. One copy of original DNA will be also there in the daughter cell. So this process is very fast and efficient and it allowing the organisms like bacteria to multiply very rapidly under certain favorable conditions. So this binary fission method is very rapid and fast. Okay, so what are the different types of binary fission? So when we come to different types of binary fission, it is always divided on the plane of division. So when certain this like amoeba is dividing, so if it is dividing like this, so this on this type, on if it is dividing on this plane, so it is dividing on the like plane of division. The division will be divided. So four main types. Simple binary fission, what is that? The cell divides into two equal halves at any plane. So it may be like this, it may be like this, like this. So on any plane it can divide. Okay, it divides in an irregular manner since its body shape is not fixed. So amoeba, we know that the body shape is not fixed and it can divide in any plane of division. Is it clear to all of you? Now we come to the second one, or which is called the longitudinal division. So longitudinal binary fission, it is along its length. So it will always divide along its length. So this is longitudinal axis, longitudinal division. And euglena, it splits from the anterior to the posterior in this longitudinal way. We will see the images in the next slide as well. But please do remember this type of example. So simple binary fission, amoeba. Longitudinal binary fission is euglena, which will divide along its longitudinal axis. It's clear? Next we move on to the transverse binary fission, that is according to the width or transversely. So transversely it can divide, that is paramecium, it splits across the middle of the body forming two equal halves. Okay. Next comes the oblique binary fission, that is they can divide at a certain angle or obliquely, neither purely longitudinal nor purely transverse. Okay, So it's neither transverse nor longitudinal, it is rather oblique. So it happens in Ceratomixa, a marine protozoon that divides in an oblique angle. So it is very rare actually. It is in Ceratomixa, it is a marine protozoon that divides in an oblique angle. So each type of binary fission ensures that offspring are genetically identical to the parent. So in all this type of division, they are genetically identical to the parent, maintaining the continuity of the species. So the species continuation will be maintained. Right. So what are the different images of this type? Simple, longitudinal, transverse and all. We will see that. Now see, in simple binary fission, what we have seen that from any part it can divide. This is simple binary fission. It is happening in amoeba. So it can divide from any plane. Next comes the longitudinal, which happens in euglena. We have told that it is happening in longitudinal part. So this like this, and along the longitudinal axis it will divide. Next comes the transverse, which is happening in paramecium. It will divide transversely along this plane. It is dividing. Okay, so this is in general I wanted to show you what is binary fission. That is division of the chromosome duplicates, and it is duplicating, the cell size increases, chromosome duplicates, and now the cell divides into equal daughter cells. So two daughter cells will be produced during this binary fission. So one cell divides into two. So this is generally the process of binary fission. This is simple binary fission. This is longitudinal. This is transverse. And the rare one, which is oblique, that diagram I have not shown you here. That is in any oblique plane they are dividing, right? So next comes multiple fission. So two types of fission, binary we have told with examples. Now comes multiple fission. So dear students, if you are liking my channel, you need to subscribe to my channel. Please do subscribe to my channel to get more and more updates on my lesson as per your curriculum, and many questionnaires, many important things I'll be sharing in the upcoming episodes. So please stay tuned, stay updated. So now let us get back to the topic. What is multiple fission? So multiple fission is a type of asexual reproduction in which a single parent cell divides into many daughter cells. Binary, single into two. In multiple, single into many daughter cells. Like this process is mainly seen in protozoons like Plasmodium, amoeba under certain unfavorable condition. Amoeba is always going for binary fission, but under certain unfavorable conditions it will go to this multiple fission. Okay. Now in multiple fission, the nucleus of the parent cell divides several times, forming multiple nuclei within the same cell. It's clear? Now then the cytoplasm divides and each nucleus gets enclosed in its own portion of cytoplasm. So just what is happening, this we can say that it divides into several times, forming multiple nuclei. So multiple nuclei will be formed, and then the cytoplasm will also divide, forming many daughter cells. So many daughter cells will form. These daughter cells are released from the parent cells, having part of that nuclear material as well. Okay. So multiple fission allows the rapid population increase and helps the organism to survive in harsh environments by producing many offsprings at once. Okay. So they help them to survive in a harsh environment and to produce many offsprings at once, not 2, 3 or like that, many offsprings at once. So what are the types of multiple fission? So though we are talking about the multiple fission types, multiple fissions does not have many classified types as binary fission, but it can be understood it is based on certain organism type and the conditions under which it occurs. Okay, instead of types based on the plane of division, multiple fission can be categorized in the following: functional or biological context. So what are that? Functional or biological contexts on which the multiple fission is being divided. Let us see that one. So first is endogenous multiple fission. So what is this endogenous multiple fission? Endogenous multiple fission is the formation of the daughter cells occurs inside the parent cell. Endo means inside, endogenous means inside the parent cells, and they are released only after the parent cell is bursting. Okay, bursting of the cell when it happens. So plasmodium, that is a malarial parasite, undergoes schizogony in the form of endogenous multiple fission in human liver and red blood cells. So they are having this, they will burst open and they will form a multiple number of cell, that is endogenous multiple fission. Now it's exogenous multiple fission. Now the formation of daughter cells takes place outside the parent body and usually in a protective structure. So they take place outside the parent body inside a protective structure. So what is it? It is actually, it is in protists and algae, which are the zoospores or the cysts, where there are multiple new cells are formed and then released when favorable conditions return. Right. So some algae and protists are there which form zoospores or cysts where multiple new cells are formed and then released when favorable conditions are returns. Okay. So cysts, they contain of new cells, and when they come out of the cell they form a new cell. Okay, so that is released when the favorable condition, then they reach to a favorable condition. Okay, then released when favorable conditions return. Okay. So here you can see the diagram of multiple fission. What is happening? This is the parent cell, and inside that there are so many daughter nuclei formed, right? So many daughter nuclei are formed, and during some favorable condition they burst open and rupturing of the sison wall is happening, and they give rise to many daughter cells. So they are giving rise to many daughter cells. Okay, so many daughter cells are formed from one single parent cell, whereas in binary fission we have seen from one cell only two cells will be formed, but here from each one they are having all the nuclear materials inside, and all the cytoplasms also, they'll form many cells from this one cell. So this is called the multiple fission process, and this is a very fast process of growth, right? So because from one, many daughter cells will be formed, right? Now, this is another special form of multiple fission. This is not commonly being spoken of. This is one special form, that is what happens there. This sporulation occurs under certain unfavorable conditions when the organisms form a thick-walled spore which undergoes multiple fission internally to produce several daughter cells, right? Now, in case of amoeba, what it happens? A protective cyst it forms outside its cell wall, and inside it will, we can say that multiple fission takes place. Okay, and when favorable conditions return, the cyst breaks, releasing many young amoebae. So now if I show you the diagram of this sporulation, you will understand what is happening in amoeba. So this is the amoeba cell. It is forming a protective cyst outside it, and inside that multiple fission takes place. So many daughter cells are born inside, and when favorable condition returns, the cyst will break, releasing many young amoebae, right? So just as we have seen in the previous one also how the multiple fission happens, this is a different form, that is sporulation. So we will see the diagram. Now here in this diagram of amoeba, you can see that this is an amoeba cell. This is the nucleus. From nucleus, this chromatin blocks are coming out, and the nuclear membrane is disintegrating, and so small this nuclei are formed. So these nuclei, they are forming with these small nuclei, several different spores are being formed inside the amoeba itself. So when they are in any certain unfavorable condition, this will happen, and when they see this favorable condition, this membrane will burst open, and these will give birth to a multiple number of cells. Like these are the individual cells. So this is the spore case, this is the nucleus, and this is the cytoplasm. Okay. So from these spores again, young amoeba will come out. So this is the way. If the spore case breaks, then amoeba will come out. The new daughter amoeba will come out. Young amoeba will come out containing a nucleus and a cytoplasm in it. So this spore cell the outer wall of the spore needs to break down. Okay, that happens only in certain unfavorable conditions, and which gives rise to huge number of young amoeba cells. So this is how this spore formation or sporulation in amoeba happens, and this is a very rare case of multiple fission. It doesn't happen regularly. Regular amoeba divides by binary fission only, but in rare cases, due to certain unavoidable circumstances, when unfavorable conditions are there, this will happen, right? So what is the difference between binary fission and multiple fission? First of all, we can say that in binary fission, two daughter cells are formed. In multiple fission, many daughter cells are formed. What are the types of division? Nucleus divides once, following by a cell division. So here the nucleus divides many times before the cell splits. So it is dividing only once before the cell division. It divides many times before the cell splits, because there are many number of daughter cells formed during multiple fission. Speed and simplicity, simple and fast process. Multiple fission is a more complex process. It occurs in most unicellular organisms like amoeba, paramecium and euglena. And protozoans like plasmodium, amoeba, during cyst formation, we can say that they are having this multiple fission during cyst formation. Okay, now favorable or unfavorable conditions now usually takes place in favorable conditions, and these usually they take place in unfavorable conditions as a survival method. I have told you this cyst formation, this spore formation, they occur in the unfavorable condition, not in normal condition, and usually these are binary fission always takes place in favorable conditions. Now, release of daughter cells immediately after cell division, but this release of cells will happen, daughter cells will happen only after the parent cell bursts open. Okay, when the parent cell bursts open, then only the daughter cells will be formed. And what is the function of binary fission? The function of binary fission is normal reproduction. Multiple fission is it is often survival from certain unfavorable conditions. So these two divisions, these all differences between binary fission and multiple fission is very, very, very important for your exams. So learn this with examples, with all these features. You should learn this so that it will be easier for you to differentiate between the binary fission and the multiple fission. So let us see what is there next. So till now we have completed about binary fission and multiple fission. In the next class we will be discussing about fragmentation. We will discuss about the regeneration, about budding and about vegetative propagation. So till this we will move on to we will study in the next class. So stay tuned till then. Say subscribe to my channel. And please do go through the full episode in detail, so that if you are skipping, you will miss many important parts which I have taught you in the middle. So stay tuned, stay updated and subscribe to my channel and share it with your friends as much as you can. Thank you. Thanks a lot for watching. Bye-bye. See you in the next episode with all other types of asexual reproduction.